The main goal of this study is to demonstrate a detailed methodology of dry root rot disease assays such as blotting paper technique and Chickpea technique. These techniques will be helpful to study the interaction between chickpea plant and Rhizoctonia bataticola. To execute the blotting paper assay, prepare Rhizoctonia bataticola fungal inoculum by scaling up from slant culture to petri plate and then to liquid culture. Prepare chickpea nursery by sowing seeds in a big pot and uproot them and dip them in the liquid inoculum and place them in blotting paper. Then, observe for disease symptoms, namely necrosis on the root rot, and leaf yellowing on 8 days after inoculation. Shown here is in nutshell of steps in blotting paper technique. To execute the Chickpea assay, inoculate chickpea meal with fungus. After incubation at 30 degrees Celsius for 15 days, Chick culture is obtained. Then, powder them and mix with pot mix. Sow seeds on control pot and Chick pot. On the following days, observe for symptoms, namely necrosis on the root rot, and leaf yellowing on infected plants 10 days after sowing. Chick pot can be scaled up and can be used for large-scale screening. Shown here is in nutshell of steps in Chickpea technique. Demonstration of blotting paper technique starts here. To prepare liquid Rhizoctonia bataticola inoculum, inoculate the 500 ml broth with a fungal agar plug from 3 days old fresh plate culture and incubate the flask in an incubator at 28 degrees Celsius for 5 days. Filter out the dark gray fungal mycelia with microsclerotia and suspend in autoclaved water and remove the media. Again, filter out the mycelia and remove moisture by blot drying the mycelia for a few minutes. Suspend the 100 grams of fungal inoculum in 200 ml of autoclaved water in a jam bottle and mix it properly. Develop chickpea nursery by sowing surface sterilized seeds in soil right. Uproot the plants on 8 days after sowing, wash them and rinse them with autoclaved RO. Take a blotting paper and fold it once and place half of it on the tray and make it wet with autoclaved water. Take plants and dip into the inoculum for 30 seconds with intermittent up and down movement. Place the plants in such a way that roots are covered inside the sheet and foliar stays out. Fold the other half and wet the paper. For control, dip the plants in autoclaved water. Keep the trays in a growth chamber for 8 days. Observe for the symptoms such as leaf yellowing, root rot, necrosis in root end, reduction in lateral root number. Demonstration of Chickpea technique starts here. Take 1 kilogram of commercial chickpea seeds. Wash the seeds with tap water and wash again with RO water and soak the seeds for 5 hours. Again, wash the seeds with RO water and fill 100 gram in a jam bottle and autoclave them at 121 pounds for 15 minutes twice continuously. After cooling down, transfer the bottles to laminar flow. Inoculate the bottles with 3 fungal agar plugs approximately 4 mm square sizes and shake it well to mix properly. Wrap the bottles and incubate them in an incubator at 30 degrees Celsius for 15 days. Observe for the bottles with dark mycelial growth around the chickpea seeds and remove the seeds and dry them in room temperature and powder them. Mix 10 gram of Rhizoctonia bataticola powder with 100 gram of soil right and keep them in room temperature for 5 days. Surface sterilize the JG62 seeds and sow them in 2 cm depth. Observe for the 90% mortality in Chickpea. 10 days after sowing. Observe for the symptoms such as necrosis in roots and reduction in lateral root number and leaf yellowing in the following days. Chickpea can be prepared in field soil as well. Impose drought stress in the Chickpea by withholding the water. Shown here are the plants subjected to control, drought, pathogen and simultaneous drought and pathogen stress. Observe for the symptoms, namely necrosis, root rot, and reduction in lateral root number and root length in combined stress treatment as compared to pathogen-only treatment. Blotting paper technique can be carried out in vitro conditions. Chickpea technique can be executed in vivo conditions. In Chickpea, abiotic stress, namely drought stress, can be imposed along with pathogen infection. Using these techniques, one can study the pathogen infection pattern. Morphophysiological changes occur during the chickpea Rhizoctonia bataticola interaction, and also screen the chickpea genotypes for resistance.